This embryology highlights screencast, Cardiovascular Development 2, deals with heart development. Cardiovascular development occurs early in the embryo when the embryo grows too large for diffusion to efficiently transfer nutrients and waste. The vessels are formed as blood islands which develop within splanchnic mesoderm. The heart originates from vessels that form within splanchnic mesoderm located anterior to the embryo, and this region is called the cardiogenic plate. Initially, mesenchyme stem cells commit to forming hematocytoblasts or blood cells. The blood islands are formed as the hematocytoblasts induce the surrounding mesenchyme cells to form endothelial blasts. The individual blood islands grow in size, they coalesce, and they give off buds. In many areas, the initial vessels will degenerate as development proceeds. At the right, we see the cardiogenic plate region. Here, vessel formation is taking place in splanchnic mesoderm located anterior to the embryo. A single endocardial tube is formed as the head process formation brings the cardiogenic plate ventral to the foregut or the pharynx, and bilateral endocardial tubes fuse at the midline to create a single endocardial tube. The endocardial tube forms the lining of the heart. Splanchnic mesoderm gives rise to the cardiac muscle fibers. These images on the right are transverse sections through an embryo from earlier and later in development, and they show how, as lateral body folds come together, endocardial tubes, which are initially bilateral, fuse to form a single endocardial tube, the future heart. The splanchnic mesoderm surrounding the endocardial tube will give rise to the cardiac muscle fibers. These images also show how the foregut, or pharynx in this case, is formed as these bilateral folds come together and create the foregut. Also notice that the neighboring coelom is brought into surround the heart and become the future pericardial cavity. On the left, we have longitudinal sections through an embryo. The detailed images show the relationship between the coelom and the heart changes. Initially, the coelom is dorsally, and then as this swings around, the coelom is ventral to the endocardial tube. This image shows an early cardiovascular system. Notice that all of the vessels are bilateral except for the merged vessels that have formed the endocardial tube, the future heart. The veins in blue drain the yolk sac and the allantois, and via caudal and cranial cardinal veins, they drain the embryo itself. This venous blood enters the heart and is pumped to ventral aorta, and the blood via aortic arches enters the dorsal aorta and out its various branches. So even as a simple tube, the heart is functioning. It's receiving venous blood and pumping blood into arteries, which deliver that blood throughout the embryo and to the yolk sac and allantois. This image shows bilateral tubes merging to form a single endocardial tube, which has begun to show regional swellings. 5 endocardial tube regions are identified. The truncus arteriosus, the output region of the heart, will become aorta and pulmonary trunk. The bulbus cordis will become right ventricle, and the ventricle region will become the left ventricle. The atrium will expand and become right and left sides, and each of those will become the right and left auricles of the adult. Meanwhile, the sinus venosus, which is a pad region in the embryo, will become 2 different things. The left sinus venosus will remain smaller and become coronary sinus, but the right sinus venosus will enlarge and become the wall of the adult right atrium. During development, then, the simple tube must transform into a four-chambered heart, all the while functioning to pump blood from the veins into the arteries. The process of forming a four-chambered heart from a simple tube is challenging to envision. We will approach it by looking at 6 snapshots of cardiac development. First, when the endocardial tube lengthens, it loops on itself, and this positions the 2 ventricles side by side, and it puts the atrium dorsal to the ventricles. This image shows the endocardial tube, atrium and sinus venosus, shown in blue, and as the tube elongates and the regions become more distinct, it begins to loop on itself. As a result, the future ventricles come to lie beside one another, and the atrium is shifted dorsal to the ventricles. This image shows the atrium located dorsal to the 2 ventricles, which of course communicate with one another. These images show later stages. The 2 ventricles are beside one another, and the atrium is dorsal to those. This is sort of a lateral view, and this is a ventral view. You see the 2 ventricles. The truncus arteriosus is the output, and the atrium has expanded so we can refer to a right and a left atrium situated dorsally in this ventral view. On the right, we have a scanning electron micrograph of a endocardial tube. It's beginning to elongate within a confined space, and thus it loops on itself. The next snapshot that we want to consider is that venous return shifts to the right side. As a consequence, the right sinus venosus becomes enlarged, and eventually it forms the right atrium in the adult. Meanwhile, the smaller left sinus venosus merges with the right one and becomes the coronary sinus, which empties into the right atrium. This image shows 3 stages of development. They're all drawn to the same size, even though as the embryo matures, this is much larger than this early stage. We can see that the atrium is colored blue, and bilaterally the sinus venosus is colored purple. The cranial cardinal vein and the caudal cardinal vein drain blood from the embryo, and on the right side they will become the cranial vena cava and the caudal vena cava. As blood flow shifts to the right side, the right sinus venosus enlarges, and the left sinus venosus fails to grow and becomes incorporated into the right sinus venosus, which is becoming incorporated into the wall of the atrium. The cranial cardinal vein will become cranial vena cava, and the caudal cardinal vein will become caudal vena cava. At this later stage, we see that the heart is enlarged quite a bit. The sinus venosus on the right side is incorporated into the embryonic right atrium. The cranial and caudal cardinal veins are distinct. Meanwhile, the left sinus venosus has merged. It is carrying blood now mainly from the heart, and it is going to be the coronary sinus joining the right atrium. This image shows the interior of the embryonic atrium at an early and a later stage. Initially, on the right side, we see the right sinus venosus being incorporated into the wall of the right atrium. At a later stage of development, this blue area, the sinus venosus, has enlarged and represents more of the atrial wall. We can see the cranial vena cava emptying into it, the caudal vena cava, and this is the coronary sinus emptying into it. Meanwhile, the original embryonic atrium is the right auricle, and sinus venosus then becomes the right atrium proper. The same process is taking place on the left side. Here we see pulmonary veins being incorporated into the wall, becoming the left atrium. The embryonic atrium will become the left auricle. The next 2 snapshots are First, the common atrioventricular opening is being partitioned into right and left atrioventricular openings by a growth called the endocardial cushion. Then we have the right and left ventricles being formed by growth and internal excavation. This creates a septum then. Originally the two ventricles communicated with one another, and as they grow they are separated by a septum which is sculpted by selective excavation. This image shows 3 stages of cardiac development. They're all drawn the same size, although the last one would be much larger than the first one. The cavity of the heart is in yellow. The sinus venosus on the right side is drawn in blue, and the cardiac wall is brown. Initially, the 2 ventricles communicate because they were part of the same endocardial tube. As the 2 ventricles enlarge and are selectively excavated, an interventricular septum is formed, except at the original opening where the 2 ventricles communicated. This process continues, forming a muscular interventricular septum, but this original opening will have to be closed by a membranous septum that will grow down from the endocardial cushion. The truncus arteriosus partition will also contribute to this membranous interventricular septum. This top series of images shows how a common atrioventricular opening is partitioned into left and right atrioventricular canals by growth of endocardial cushions. The bottom series of images show how excavation of a ventricle produces papillary muscles, chordae tendineae, and cusps of the atrioventricular valves. It's quite remarkable then that these complicated structures are all formed by selective excavation of the growing ventricular wall. The next snapshot of cardiac development is the formation of the interatrial septum and the foramen ovale. In utero, the right atrium receives much more blood than the left atrium, and so a foramen is necessary to allow blood to flow from the right atrium to the left atrium. But at birth, a valve is needed to close that opening. Formation of a foramen ovale involves 2 septae and 3 foramina. Below, we can see the first septum grows and closes the initial communication between right and left atria. But before it closes that opening, a secondary opening must develop by fenestration of that primary septum to allow blood to flow from the right to the left atrium. And then a second septum grows down, but that septum never completely closes off, and so there is a foramen ovale formed. And as we see here, blood then can flow from the right atrium through the foramen ovale through the secondary opening. When pressure is equal on both sides, this septum can act as a valve closing off the foramen ovale, and eventually it grows together. This image presents a colorful view of interatrial septum formation, shown in section on the left and as viewed from the right atrium on the right. Here we see a septum growing down, about to close the original opening, and fenestration, a breakdown of the wall of that septum, allows a secondary opening to develop, and that's what we see here. So as this first septum closes down, the secondary opening allows blood to flow from the right atrium to the left atrium, and meanwhile a second septum is beginning to grow. And so that's what we see here. This is the second opening that allows blood to flow, but the second septum, colored in blue, is beginning to develop. And here we see that an opening has persisted in that second septum. That opening is called the foramen ovale. First septum can act as a valve to close that off when pressure is equal on both sides. This is a surface view of the foramen ovale, an opening in that second blue septum, and how the first septum can act as a valve, preventing blood from flowing through the foramen ovale when pressures are equal bilaterally. Notice also in these two views that the interventricular opening is closed by a membranous septum that goes between the endocardial cushion area and the muscular septum that's developed by excavation. The aorta and pulmonary trunk are formed by a petitioning of the truncus arteriosus. The petition occurs in a spiral pattern, and as a consequence, the pulmonary trunk and aorta spiral around one another. That spiral petition also contributes to the closure of the interventricular opening with this membranous septum. The aortic and pulmonary semilunar valves are formed by selective erosion. So here we see that process. This colorful textbook image illustrates petitioning of the truncus arteriosus. On the right here we see that the pulmonary trunk and aorta spiral around one another. This diagram indicates that that is a result of a spiraling partition. On the left we see 3 stages of development. Now this process is very difficult to envision, but it begins with 2 ridges that grow in from the sides of the wall of the truncus arteriosus. And as these ridges grow in, they spiral around one another as indicated by the arrows. And the end result then is a pulmonary trunk and aorta that spiral around one another. That spiraling enables the pulmonary circulation to be in series with the general body circulation. Were they not to spiral, the 2 circulations would be parallel to one another. This image illustrates semilunar valve formation in the aortic and pulmonary valves. Initially, the truncus arteriosus is not partitioned, then ridges come in to partition it, and here we see an aortic valve and a pulmonary valve, and there are 3 semilunar cusps that are formed. Below, we see that these growths are selectively eroded. If the erosion is too excessive, then there will be a valvular insufficiency where they don't completely close and there's backflow through the valve. Or if there is too much growth, then there will be a stenosis and the lumen will be constricted, and this will offer excess resistance to blood flow. A cardiac anomaly that occurs in a number of species is the tetralogy of Fallot. Fallot is a man's name, and tetralogy refers to 4 defects. All of the defects are related to defective spiral septum formation within the truncus arteriosus. This image illustrates the tetralogy of Fallot in a surface view and in a section through the heart. A basic problem is unequal partitioning of the truncus arteriosus results in a stenotic or narrow pulmonary trunk and an enlarged aorta. On the right, we can see all 4 defects. The stenotic pulmonary trunk is evident. The enlarged aorta actually overrides both ventricles because the septal defect failed to contribute an intramembranous septum that exposes the right ventricle to the high pressure of the left ventricle, and as a consequence it hypertrophies. So we have 1, 2, 3, and then the hypertrophy is a consequential 4th defect. So this concludes this embryology highlight screencast dealing with heart development.